Okay, so this is the abstract of the paper that we're going to talk today. Here, what they are doing is this is a literature review. So rather than uh, showing a lot of experiments, yeah, what they're doing here is something different, yeah. They're collecting all the information from previous paper and summarize it in a way that is accessible for everybody to understand and to show the all the summaries of finding and potential new hypotheses that they are trying to develop. Okay, so uh, here, this is the abstract. As you can see, this you can find, and I will in a previous video I already show you how you should do something similar for your literature review and explain and try to copy some of the style or trying to learn what they are doing. Yeah, here if you see there is no experiments, they are just summarizing all the data that happened here. Okay, so again, so as this is a review paper, the first thing that you have to is skimming all the paper, look for the headings, look for the information, look for the figures, scan it very fast, not for the details, but to know if this paper actually is important and you need to commit, and to what degree you need to commit to learning these things, to or not to learn, yeah, to read all the paper in a lot of detail, and to what degree you need to understand all the details, yeah. If this is going to be, if it, you need to add one section for your review paper, or you need to go and understand it in a lot of detail, yeah. So I'm going to go through the figures, and here in this figure, what they are showing you, the key of this idea of this slide is that to show you that they found that the direction in which cells are extruded has important consequences for its fate. So in the previous classes, we talked that if the cells are invertebrates are extruded basally, they can be killed because there are signals that allow them to be killed. And also in this paper, they're going to focus more on what happens if the cells go in the opposite direction, that they can bypass uh, this type of cell death, and that way they can uh, lead to the progression and invasion of cells and produce cancer. Okay, so again, in the first paper, what they describe is generally the normal process, and in the following papers that they discussed, they were focused on the applications to cancer. And here, I'm just going to give you a review that is going to be very general from the paper. So generally, review papers, they talk in a more general way of what is happening in other in the field. Okay, uh, what they introduce in this idea, in this figure, the idea is that cells are extruded and removed from the epithelia. If they can die, they can be removed alive or dead, as we have described, by as a response of crowding during homeostasis, during normal processes, or by apoptotic stimuli. Yeah, so there's a type of cell death here, uh, oikos, oikis. Yeah, so this type of cell death, remember when you have a or an in a word in Spanish or in English, uh, it means without, yeah, or absent of something, and this oikis is house, home. Okay, I made it. Yeah, good. So this cell, what they are saying is this type of cells is because it doesn't have a house in the sense that it was removed from the normal epithelial, so it's in the not in the proper place, so it doesn't receive the correct signals that keep them alive, and don't if it's not in the correct place, it will die by this type of cell death. Yeah, so well, other stimuli. Well, the other one died. Sorry, here there was a two. Well, other ones died for a different reason, yeah, because there were spaces and there was a lower or the cell machinery work in a different way. Here, what is happening is that they are first removed, and then this lead to the process of apoptosis, but it's a slightly different way of apoptosis. Okay, so this is this very weird work, and here this is what they are talking, that they are able to bypass. They are not anymore in the epithelium. They should be dying, but as they are survival signals in the stroma, they are able to survive. Okay, so this is the first figure, and I want that you, in the previous paper, we just talk about that the cells go up or they go down, they go apically or they go basally. And here we will talk about what happened with the cell biology. So here we will talk about things that you have discussed in our classes, and I try to remind you of some of them here. So for example, actin is very important. So remember, actin is a protein that is in charge of uh, everything related to force, to mechanical forces in the cell, to changing the shape, to allow the cells to move in different ways, to the cellular oscillations that we talk. They generally happen in actin is involved in all those things. So here, actin is changing the shape here and is allowing the cells to detach. Yeah, so this actin is very important. So what we are going to do is look what happened in normal conditions. Then we are going to see how these normal conditions are disrupted, what happened with actin here to do those things. Another thing that is very important that they didn't talk in the previous paper is about cell junctions. So again, if you remember, these cell junctions are attached, are linked to the cytoskeleton, in this case to actin, and they are also, this one will be attached also to the nucleus. Yeah, so there will be a communication from the cell adhesion to the nucleus to change the gene expression. And here the cell junctions, if you see, they need uh, they the cells to be extruded, they need to downregulate the cell adhesion, and they need to later so detach to allow the cells to go out. Okay, so in this paper we will be talking about all these processes. And just to remind you, this is, here we are talking about epithelial cells, that all this that we are talking about junctions and going in the stroma or in the lumen is mainly about epithelial cells. Okay, so now we're going to talk about in more detail. So the first time that you scan the figure, really quick, you didn't notice anything of the detail. You said, this is a paper that I need to read, so I will go in and see all the details, yeah? Or to see if this is a paper that you want to include in, and you need to understand all the details, you can read it in a lot of detail. So, for example, here you notice that there are and I am going to focus on the following slides in three main sections. So here they look blue arrows, they show you something, that there is cell death, a type of cell death that we talked in the previous slide. The second one, this is not showing the first paper, that they think that this could be a tumor suppressor mechanism, this type of cell death. And again, the third one, they're going to, during all this paper, trying to show you what happened with the actin, with the myosin, with the cell adhesion to produce, describing the normal, in what happened in normal condition and what could be happening in abnormal condition to be able to extrude not apically but basally in vertebrates. So let's start. So we know that typically the live cells extrude apically and they go into the lumen. And how this happened? So because they have this contracting and intracellular actomyosin band that is located basolaterally to squeeze the cells out. So remember, here you have, here is the apical region, here is the basal region, and in this area is this region here is closer to the basal, but this is the basal lateral region. That this is where the actin is going to be located, that is going to reorganize. In the other ones is located in this region with the actin. Here it's going to move and it's going to squeeze out the cell outside. Okay, and this is something that is not only shown by them, it has been shown uh, for several decades. What they are going to do is um, that they're going to describe what happened with during cancer with tumor suppressors and oncogenes. Uh, by what happened with this mechanism that we didn't cover in the previous class, but they did many experiments. And what they are saying is that, you know, when the cells are extruded apically, they start losing survival signals, and they died by this process, this specific process of cell death. Okay, so far this is more or less similar to what we hear before. However, there is something slightly different. That is, generally we talk about that they as they don't have the signals, they died, but they say something else. So what they are talking here in the with the red arrows is they are describing that this could be a tumor suppressor mechanism. Why? Because imagine that you have cells that have upregulated survival signals. Yeah, so if you have these signals that are bad, yeah, that generally you have some cells that have correct signaling. However, if these signals are not occurring in the right way, if the cells is extruded later, as it's going to be in the lumen, there is no extracellular matrix, yeah, that it will allow them to continue those type of signals. They are not going to be surrounded by other cells, so they're going to start losing those signals. So removing those cells into the lumen, it could become a tumor suppressor mechanism. Yeah, and this is what something new that they describe in this paper. Uh, Now I'm going to show you that this is the center of this paper is talking about what happened when the cells go basally. Okay, so something that now they want to focus is, now we already show you what happened in the normal conditions. Now let's focus on what is happening here in the abnormal conditions. So in the abnormal condition, you will have, we know that this process happens less frequently, yeah, and they show in other papers. I just told you that they talk about that, but I didn't go into a lot of detail, and you can read the papers. I'm just going to tell you what they talk in the review. So they, here again, they don't show experiments. They show the results in a schematic way, what they in a more simple way to what it happened. For your review, you know, in literature reviews, you generally don't cite the review paper. You go to the original paper. You can read the literature review and then cite go to the review and to the original paper and cite the other paper. Yeah, this can give you just the the simpler version to later go and read the other paper. Okay, so in this paper, what they are going to show you is, okay, but what is happening with oncogenic mutations that they died, that they, that they extrude apically, that they are able to bypass this type of cell death, and they are able to escape from the epithelium. And I remind you, they have a lot of survival signals that the cellular matrix is going to amplify and is going to help them. However, here they haven't tell you still what is happening with the actin and with the cell adhesions, and this is what they are going to tell you later in the following slides. And they can also, using this, they can propose what type of mechanisms that we see normally during cancer can be linked to this process of cell extrusion. Well, in the previous case, if they go to the lumen and the cells have a mutation that allow them to have to upregulate the survival signals, and in the apical in the lumen they will going to die if they extrude basally, and you have the mutations there that are upregulating survival signals. What is going to happen is that they are going to have the stroma. They're going to be in the extracellular matrix that they can continue amplifying these survival signals. Okay, and as we talked in previous classes. So they're able to bypass this type of cell death and they can escape from the epithelium. And what they propose in this paper is that, and I want to emphasize this, be very careful in your papers, look at what they rate. This may provide a novel mechanism. Okay, so this is an area that is still grow and that is still in development. So there is a lot of things that it seems to be it seems to be fitting to a new mechanism, and they are trying to be careful because it required that there will be more papers published to describe all this information. But the idea is that these novel mechanisms can upregulate survival signals and initiate uh, tumor invasion or all these things that we have to study in the beginning of the course that can initiate cancer. Okay, so in the following slides, I'm going to show you how tumor uh, examples of an oncogene and a tumor suppressor. Yeah, what happened with this type of cells that have those mutations with the actin, with the cell junctions, and how they can be squeezed out of the epithelium. So to understand how this process happened, you need to first remember what is this S1P. So I know that we have talked a lot about hundreds of molecules, and this is another type of molecule. I have been receiving some of your review papers, and uh, you think that I am talking about too many pathways and molecules. When you're going to work in your paper, you will see that you will have some of the pathways and some new molecules. So you are living in an exciting time that we are figuring out all the circuits in of cell biology. So um, so it's very interesting, but at the same time, it's a little bit overwhelming. So again, don't feel overwhelmed. I'm going to try to summarize what is the function is this S1P that is a lipid, yeah, that works as a signaling molecule and that is involved in this process of basal extrusion. And why this is so important? Because it has been shown that there could be modification in the signaling of this S1P in oncogenes and tumor suppressors. Okay, so what is this S1P? So this S1P is a bioactive lipid mediator that is a group of molecules that independent of the tissue. It is involved in signaling in different signaling pathways, but in different tissues it will have different signals. Here, for example, I show you a schematic took from this paper here that is describing what happened with in glioblastomas, how the modification of S1P is going to activate different pathways. Okay, so important things here. Again, this is a signaling molecule that it has a receptor here, and if you see it says 1 to 5, again, I just make it bigger, that small part. So there are multiple receptors, yeah, depending on the tissue, yeah. So for this one, they will talk about for the extrusion, they will talk about the S1P2 receptor, yeah. And okay, so something, so again, that you have listened hundreds of times. So you have this ligand, this molecule, signaling molecule that it will attach to a receptor. And what is going to happen? The same thing that always happen, that it will join to that it will go to a transduction in the of the signal. And how it does this transduction? Because it activates a G protein. Yeah, that is always the same story. And here you find all friends that we have been talking all the time, RAS and the MAP kinases pathway. That here you start seeing again and again the same circuits that are happening. So here these are invading it. They are involving invasion and proliferation. Yeah, and we haven't talked a lot about Tor, but maybe in your paper you will see that they will be talking about Tor. This is an OR pathway that is very important for many functions in the cell, and here in this case it will be for angiogenesis and survival. Yeah, this is here also an old friend, the JNK pathway involved in cell death, and here another old friend that maybe will be appear in your paper, that it will be involved in the migration. Again, there is so many things. I don't want to tell you all the details. I just want that you remember that this is very similar. You start finding the same process. That is the general idea: ligand, receptor, transduction, and you activate or deactivate some pathways that you have heard multiple times. Okay, and you will see some process that are important in different type of cancers. Yeah, some circuits that are important. So it's not a surprise that this is that has a G protein and that here could be involved in RAS, in TOR, in the JNK pathway. Here, oh, the MMPs. Yeah, so again, all friends that with time you will start memorizing them. Okay, so now we're going to move into the figure number 2. Now that you know what is this signaling molecule and that it has a receptor. And here they start by showing you what happened in the normal cells. So in the figure 2, they are going to show you how cell extrusion can occur basally, and they start by showing you what happened in normal cases. So yeah, so in addition to talking about the actin, they're going to show you what happened with the microtubules. Okay, and so key things here, they are not only going to talk about the cell that is going to extrude. In a normal situation, you have one neighbor cell, another neighboring cell, and here the cell that is going to extrude. Okay, so I want that you see, try if you want, you can stop the video and try to see, write in your paper to send me in Moodle this, yeah, as an in-class assignment that I know you were paying attention to the video. What do you see that happened between the initial signal, the initial time that they said this cell has to extrude, to the final time? Yeah, what happened with the microtubules, with the SIP, with the receptor, and with the actin? So try to give a look. Okay, so I'm sure that you stopped the video, that you wrote all the things. Now I'm going to show you what they talk in the paper. They said that in the wild type is not only the extruding cell that the one that is playing a role, is the neighbor cells that are helping that to properly be extruded. Yeah, and how they do it. And let me delete this. So in the initial time, what they do is the microtubules need to be reoriented. And if you see, they are first on the top, and then later they are moved to the basolateral. So again, apical, basolateral. So they move to the bottom. And if you see, they are not the only one that are moving. All the structure is moving to, in an organized way, and start pulling, taking the cell out completely and helping it to extrude. Okay, so Microtubules are very important, and the neighbor cells are also important. Okay, so now we're going to move into more detail what happened with this with the S1P and S1P2. So we know that already the microtubules need to reorient it, and now we're going to think what is or they are going to this is what they assume that is happening. So if you see here, these molecules, the molecules and the receptor are located in the apical to basal side, and what they are going to do as the microtubules reorient, they are restricted, they are confined in the basolateral region, and if you see again, the neighbor cells are very important. So this the receptor um, the receptor and the S1P of the neighboring cells are going to trigger the signal that say you need to extrude to the other cell. Okay, so again, the microtubules are going to produce a change, and that so the neighbor cells are very important, and they will end up triggering the cell extrusion. So what I show you is you saw in the previous slide that the changes in the microtubules, and then I show you that there was a change in this molecule that is called the S1P, that we think that it possibly the most likely is that it is restricted, it is confined to the basolateral region, and in that region you also have the receptor that is expressed again in the neighboring cells, and this will trigger the extrusion. Okay, so we already talked about what happened in the microtubules, reorientation, then is this move to the basolateral region, this S1P and its receptor, and this here again, we are not only talking about the extruding cell, but what is happening is that the tissue is helping the cells to extrude. Okay, so now the final part that they describe in this figure is what happened to the actin. Okay, so what is happening to the actin here is that I want that you give a look to what happened to the actin there. Yeah, so it was actin was located from the apical to the basal area. Yeah, uh, in the border in the boundary between the two cells. Then it got restricted in that area. Yeah, and how this happened. So later we need to think about the potential molecules that are affecting actin to do this kind of things. Yeah, and that generally are GTPases that are again molecular switches that are doing all those things. So again, what I want to give you is I don't not all of you are cell biologists. I just want to give you the very general idea of what is happening, and what is happening here is microtubules are from the neighbor cells are rearranging all that is happening here, and in this case there is a change in the actomyosin uh, molecules, and there is a constriction here that it moved them to the basolateral region, and possibly this is reinforced by a molecule that is called Rho. Okay, and I'm just going to go into that Rho in a second. So we talked in the previous slide about Rho. Now I'm going to just to remind you, because I know this class has so many things. And here, if you remember, we talk about molecular switches. So here I'm trying to draw a very ugly switch that this is, but I hope that you understand. So we talk about switches that turn on and off pathways depending of if they are in they have the GDP or GDP form. So depending on that we have covered before, and we talk about many small GTPases, and one that is called Rho is important in determine an activator or turning on or turning off the changes in the cytoskeleton. So this row is involved in the extrusion, yeah, because the cytoskeleton need to reorganize to extrude the cell. In the apical oscillation that we are studying, also very important, and I'm going to talk about that now. They are also involved um, in movement. So if you disrupt row, uh, the cytoskeleton is going to start acting in a different way, and it can lead to invasion, it can lead to wrong extrusion, it can lead to atypical patterns of oscillation. So as you see, this is involving a lot of process. Here I just wanted you to remember two things. So this is the theoretical aspects, and here is more the practical aspect. So in our way, if you understand this, yeah, you understand how the cell function, and if you understand this later, you know which molecules you need to create a mutation to test your hypothesis. Okay, so this is why we give you so many molecules. I don't want that you remember all of them, but if you want to do an experiment, if you need to understand both components. Okay, so here what they are showing you is that in A, in this paper, what they are describing that this this row pathway is the actin and myosin, yeah, is a mechanochemical pathway, or there is a mechanical chemical signaling that is controlling how the actin and myosin should be activated or deactivated. Yeah, in other words, the forces of the cells acting here and acting outside are very important, and they require both a genetic component, receptors, and also it required that the bioelastic properties of the cells, how much, for example, in plants, how much water they should be. If they have less water, it should be another property. In animal cells, water also play an important role, but there are many molecules that are involved there, and they have two components. So you need to understand that these two work in parallel, the mechanics and also the chemical and genetic components. Okay, so if you understand that, now you understand that as we have these small GTPases, in this case row, that require this GDP and GTP molecular switch, yeah, this will activate other proteins like Brock. You might find it in your paper. Myosin, you might find it in anything related to the cytoskeleton, and based on that, you will have changes that are involved in the here that they describe as the internal forces and the external forces of the cells. Okay, so to summarize this idea, I told you about Rho, that is an important molecular switch, and that Rho and all this actomyosin is a way, is a mechanism that is not only mechanical, or it's not only genetic, it's not only chemical, it's the interaction between these two. And in the case of the oscillations in cell area that we are studying in your projects, they occur because the coupling between the actin and the myosin driving the formations. So this is why the cells can become bigger and then smaller, and they can become bigger and then smaller, until eventually they said they died in some cases, yeah. Or they can, in some cases, they can continue growing or getting smaller. So disrupting this role, it could be important to understand why some cells died and why cells don't die. Okay, so this is important. Another important point that I told you is that they require both chemical and mechanical, and here the they should be a passive component of that. So cells are deformed because there is external forces that, and there is the cells, even if you squish a sponge, with your hands, you will see that they passively get smaller. Yeah, so here this is what they show with the spring and the dash pot. Yeah, that those things are happening, but at the same time, cells are different from a sponge because they need to produce a change that is not passive, that is active, so they can respond. And in the sponge, if you release your hand, it will eventually recover. The cell need to have a better response. That is an active movement. That is a change in the cytoskeleton. Okay, and is dependent of the density of myosin. Okay, so these very vague parentheses to remind you something that we will use and you might use in your papers, and that is very important in cell biology and in all the papers that you might read. That here what I told you is that there's a biochemical model in which, depending on the amount of tension and in the gene pathway, here you can change the gene expression and change the regulation of the tension in the cell. Okay, so now I'm going to continue back into the class, into the topic of the review paper. But remember, rho is very important for for this process of extruding basal the cell. And there is, I just say rho in the class, but there is, again, as usually, if we zoom out, there's many molecules involved into that. Okay, so now that we understand what happened in normal cells with the microtubules, that will happen with the cells with these with the S1P and with S1P receptor, and now that we understand what happened with the actin, so I'm going to show you here in this slide the wild type, but also what happened with a tumor suppressor, that in this case is the APC mutant. And while this one, in the wild type, remember the cells will extrude apically, this one the cell will extrude basally. So here I want that you stop the video and try to see based on this schematic, and again write it down, and then we can talk. You can upload it on Moodle, and if you're in class we will talk about this. So similar to the previous example, I'm going to go to each one of the molecules. Okay, so here I told you that these are mutations in tumor suppressors. So you remember they are the checkpoints of the cell cycle. So the cells now, they are cells that are not supposed to keep dividing, that they have problems there, they will keep doing it. So in this case, they describe what happened with the APC. That is a common tumor suppressor that they describe, found in many tumors. And what they said is that if you see, there is a disruption in the microtubule dynamics. And here, key thing that you need to remember, well, the cells here, they require their neighbors. So neighbor 1, let's put it neighbor 2. Yeah, now the cell is going to start acting autonomously. So yeah, they have the microtubule dynamics start changing. They start not the other cells. They start not responding to what other cells are doing, or yeah. So here the cells are not working as a community. Okay, they are start working by themselves, doing something different. Okay, so let's start with what the microtubules are doing. So here, what you saw before is that they reorient and they start moving the microtubules into the basal lateral region. Here, the microtubules are not reoriented, are not attaching to the other cell. So as a result of that, there is no reorientation. The other molecules that we are going to talk about, the S1P and the receptor and the actin, is not going to move to the basolateral region. Okay. So I already told you what happened with the microtubules. I already told you what happened with the S1P and the S1P receptor. And I show you that, there is a reorganization of the microtubules, and these two guys stay below here in the basolateral region. Here in the tumor suppressor, there is no reorganization. So there is not a real reorganization of these molecules either. Yeah, so they are everywhere. So again, the cells start acting individually, start having their own behavior. And now look what happened with the actin. The actin, instead of being all over the from the apical to the basal region, it start locating only in the apical region, and without the help of the microtubules, without the interaction of the neighbor cells. Again, the key word here is autonomously. Yeah, start creating a constriction in the cortical region, in the apical region of actin and myosin. And as you can see, this constriction is completely the opposite to what it was happening here. And now, as it's constricting apically, the cell will go basal. Yeah, and key thing, this seems to be very atypical for vertebrates. However, in uh, in flies, in the epithelial, we generally study in flies. This is how things happen in flies. That there is a cortical actin and myosin band that it is going to uh, constrict the cell apically, and the cell is going to be removed basally. But for vertebrates, this is completely atypical. Okay, so I show you what happened with tumor suppressors. Now I'm going to show you, in this case with this APC mutant, now I'm going to show you how with an oncogene it also can go, it can extrude basally, but it modifies the same components in a slightly different way. Okay, so let's get into that. Okay, so now we are going to talk about this mutation in RAS, and that this will disrupt the apical extrusion. So let's focus on, I want that you observe the figures and you tell me what is the difference. Is the difference in the microtubules? Is the difference here in the S1P, or is the difference in the actin? And again, write it down. Stop the video and write it down, and you have to tell me, upload it on Moodle and tell me what is the difference. Okay, so definitely microtubules here. If you remember, there was a reorientation from the neighbors, yeah, that it caused the cells to uh, move the S1P and the S1P receptor, yeah. And if you see here in the KRAS or the S1P mutant, what you observe is that there is a reorientation definitely from the microtubule, so this is fine. However, if you see, there is no S1P or S1P receptor. Yeah, so here, although the microtubule is doing their job, there is a disruption of the normal mechanism, and as a result of that, also actin is also not located everywhere. It's only located basally, and it will autonomously again constrict there. So and the cells are going to go in a different direction. Okay, so what I show you is that, well, here there was the reorientation of the cells. Let me delete all these things. Okay, so now that I delete most of the things, I show you also that, so now the receptors are here, and there is an interaction between neighbor cells and the extruding cell. However, when you have a mutant for this S1P molecule for the receptor, or you also, or you have this mutation in RAS, what happened is, although there was a reorientation, you don't find the other molecule. So There's a disruption in the apical extrusion because there was a downregulation of both the S1P and the S1P receptor. And so if there is not this type of signaling, what is going to happen is that there will be actin and myosin will create a constriction apically instead of basally, and the cells are going to live in the opposite direction. They're going to go out like this, extrude like this instead of extrude like this. Okay, so if you see it's not exactly the same thing here, is because they don't have S1. So this is okay, but now these two are the ones that are not okay. In the other one, it was mainly the microtubules that it was not working, and it was causing that the other ones, it was causing a chain effect based on what they describe in this paper. Now I'm going to talk about where this S1P and S1 and the S1P receptor went, and I'm just going to talk a little bit about autophagy. Okay, that this is a very important process of the cells, and then I will tell you what happened, why these guys are not anymore, why these lipid and receptors are not there for the signaling. So now we're going to talk about autophagy, and if you remember, auto and phagy, this if you study the roots, this it means the in self, and phagy or phagos is eating. So it's a cellular process that allows a cell to to some degree eat himself, but the idea is that the cell recycle the materials, has a process to recycle their own materials. Yeah, so the autophagy is a natural process by which the cells break down. So if you have an old and damaged cell, an unnecessary cell, or a dysfunctional component, the idea is that the elements of the cell, they are still useful. All these ATP, all these amino acids are useful. So the cell is going to use enzymes, yeah, all these different lysosomes, all these different organelles to reuse those materials. Yeah, so it's able to repurpose those components to fuel and build or maintain the cells. Okay, so pretty simple. So it's not actually that he's eating himself, it's actually that he's using the old things and recycling this, and it goes through 4 different stages. So sequestration, so you have this autophagosome that is formed around the cytoplasm and organelles. So here you see how the phagosome, autophagosome, it found the places of the cell that are old or unnecessary. Then all these sections, so for example here, these mitochondria, these different organelles fuse with the lysosome, and if you remember, the lysosome is full of enzymes. Yeah, so here you see later it goes to the degradation. So here the lysosome is going to release enzymes that are going to break the components there, and finally the lysosome is able to reuse, to break those components to later reuse them for different metabolic pathways. And in Cells and Energy, we will talk about each one. Cells and Energy, no, sorry. In cell biology, we we talk a little bit about this vesicles and all the different functions of the cell, and there is again they have found so many things in the last years that I only give you a small portion of what they found, a um, very general view of this topic, but it's definitely very interesting. Okay, so why autophagy is important? Because the S1P and the S1P receptor, they in this mutation, they have gone through autophagy. So let me show you that. So I was telling you that KRAS oh sorry, KRAS and S1P mutant. They do something similar, that is the cells extrude apically. And why? Because the receptor of S1P and S1P are downregulated. So what happened with these two molecules? So that autophagy degrades them. And a puncta, when we talk about a puncta, it's always when we make images and of stainings, puncta refer that you find the molecule, the that structure, or disassemble, or just in vesicles. So you just find puncta there, and are like the key materials that are going to form something. So this is when they talk about puncta. So they it seems to be that all those S1P seem to be removed by autophagy. So how do you know if autophagy is responsible for this, for destroying this thing? You just go and make an experiment to remove autophagy, and when you remove autophagy, you find that the apical extrusion can be rescued uh, if you remove the autophagy. So it seems that with KRAS, you can still fix this. So, and they can go and die in the right way if you um, fix the mechanism of the autophagy. So this could be a potential treatment for some of the type of cancers that is unexpected, according to what they described. That you know, removing the autophagy is going to solve this problem. However, if we disrupt autophagy, there will be also other problems. So it has to be, again, in a control and very target way if we want to try to do things. So for a treatment for patients. So, but this is a very interesting finding. Okay, so I show you uh, two things: how both cell extrusion can appear in tumor suppressor and in oncogenes, and I show you those very two examples. They could be more different mechanisms, but what they describe here is that they can use different mechanisms, but both use the actin is apically constricted, and as a result of that, the cells are going to go down. Now, I'm going to, there's a lot of more information there that if you're interested in this topic, you can read it. I'm going to move into another topic, so the class is not too long. And I'm going to show you the role of this invasion, sorry, of this basal extrusion into process like invasion. Okay. Okay, so in this slide, I wanted to show you the role of this basal extrusion into invasion, yeah, tumor invasion. So here, if you see, there are two main mechanisms. One, where you have basal extrusion, yeah, that the cell is escaping individually. Yeah, this is one mechanism. And if you see, there are surrounding cells. Here, there is still a coherent, but the surrounding cells are not are not the are normal cells. Well, here the only one that is escaping is the basal cell. Here you'll find all of them. That is the second mechanism, that you have all of them are transformed cells, and what they think is that they can escape basally too, and as they have still adhesion molecules, they can pull the other cells. Okay, so I show you that there are two mechanisms. Yeah, that they said that these transform cells that are in blue, that when they can invade basally, and they can do it by being individually in a tissue, yeah, surrounded by wild type cells, what other type of cells, or if you have many transformed cells, they can also display a different behavior, and they can possibly escape all of them together. So let me show you the mechanisms that they propose. So for the first mechanism, so to escape the cell, if you see here, there is not they don't show in this schematic any coherent. Rather than saying that there is no cell adhesion, what they are saying is that there is a downregulation of the cell adhesion, and the cell is downregulated. The adhesion is downregulated. It allows the cells to detach. Yeah, it's like you remove from a wall a single brick. Yeah, and you can remove it there. And so as these cells lose contact with each other, they migrate away as a single cell. And if you see, these ones are still, they repair the wound or the hole and they are attached. Okay, so this is one way that they propose in which tumor inversion can occur by just escaping one of the cells by downregulating it here. Now let's go into the other example. So here you have in example B uh, something that is very different, in which the coherent is still present everywhere. Yeah, and instead of having one single cell, you have cells both, or in the epithelial, still in the epithelial and in the stroma, or mainly leaving the epithelium and going to the stroma. What they are trying to show here is that the coherent is still retained. Yeah, although they they escape, they basically are extruded, they retain the coherent, and as a result of that, all these cells work as a group, and they are able there to proliferate and migrate together. That we have seen both individual and collective migration. Okay, so I hope that this summary of this entire paper, of this review paper, has been useful. I just show you uh, how this basal extrusion, according to these authors, can be important. And remember here, as this is a review, I didn't focus on the details or the experiments. I just tried to make as clear as possible with the schematics the different processes that are taking place. There have been many more reviews about these topics, and there will continue being more reviews because this field keep growing faster and faster. Hopefully, this was useful to write your literature review. And if you have any questions, just let me know. Have a good day and bye.